In this second part of B1.1, we want to turn our attention to lipids, otherwise known as, or more commonly known as, fats. But lipids actually include fats, oils, waxes, and steroids. And these are all substances that are hydrophobic, which means that they don't interact with water. They're not necessarily repelled by water, but they just don't mix, and they're not attracted to other nonpolar substances. So hydro meaning water, phobic meaning afraid of or a phobia of, and so they don't mix with water. And these are substances that actually dissolve in nonpolar substances such as ethanol and acetone. For each of the different types, oils, types of lipids, oils, they solidify at a really low temperature, so they're typically liquid at room temperature. Fats specifically are solid at room temperature. Wax liquefies at high temperatures, melts like a candle wax. And then steroids are unique in that they have a 4-ring structure. We'll look at some examples and some shapes of steroids a little bit later on here in a moment. The first molecule type that we want to look at is called a triglyceride. And this is a molecule that combines 3 fatty acid tails with 1 glycerol molecule. And a glycerol is a naturally occurring alcohol. It's an odorless liquid. It's oftentimes used as a solvent or even sometimes as a sweetening agent. It can also be used in medicine. And so triglyceride, means that it's got 3 fatty acid tails that are attached to this glycerol. They're connected, not surprisingly, maybe same as what we saw with carbohydrates, by a condensation reaction. And again, specifically, this is dehydration synthesis in which water is removed, an OH and an H is removed to be able to make that connection. And this forms an ester bond. The acid reacts with the hydroxyl group, the OH, in the alcohol. The molecule itself is entirely hydrophobic. It doesn't interact with water molecules or water-based molecules. And the triglyceride molecule can be an oil or a fat depending on what type of fatty acid tails it has. The triglyceride molecule is similar to another molecule called a phospholipid, but phospholipids have just 2 fatty acid tails with a phosphate group instead of a 3rd tail. And that phosphate group is kind of the head portion, and then the tails are the 2 fatty acid tails. And this molecule is unique in that it's partially hydrophilic. The head portion interacts with water, but the tails, as the fats, are hydrophobic. And we'll talk more about phospholipids when we get to membranes in a future coming unit. And looking at the structure of those fatty acid tails, it's really an unbranched chain of carbon atoms that has hydrogen atoms that are covalently bonded to it. The acid portion is a carboxylic group, and it's followed by a methyl group at the other end. The length is variable, but it's usually 14 to 20 carbon atoms in total. These fatty acid tails can be either saturated or unsaturated. And when we say that it's saturated, that means that it's fully saturated with hydrogen atoms. So the carbon has all single bonds to those hydrogens. It's saturated with hydrogens. Unsaturated results from a double bond between the carbon atoms, and so this removes some of the bonds that are available to form with hydrogens, and thus it removes 2 hydrogen atoms. And so the carbon atom is still making 4 bonds, but 2 of those are to a carbon, and then the other 2 are a hydrogen and a carbon as well. With this, in our unsaturated, we can see the fatty acid tails be either cis or trans in their orientation of structural arrangement. The cis structure is the most common arrangement, and this produces a bend in the hydrocarbon chain, and it makes the molecule less good at being able to pack together. It can't be compacted as well, and so because of this, it has a low melting point, and it's usually liquid at room temperature. And so this would be cis fatty acid tails. A good way to remember this or think about this is cis kind of sounds like sisters, and sisters would be on the same side. They like to be close to one another. Trans fatty acid, the structural arrangement of that is trans. They're on opposite sides of one another, and so this does not produce a bend in the hydrocarbon chain. It's a straight chain, and as a result, it has a higher melting point, and it's solid at room temperature. And generally, this is produced artificially. Margarine and processed foods would be examples of foods and compounds that have trans fatty acids. And according to the FDA, these have some serious negative health implications. It would be best to avoid foods that have trans fatty acids. Triglycerides as a molecule provide a lot of energy storage both in plants and animals. For animals, the energy is stored in adipose or fat tissue. And humans, this is typically found beneath the skin as well as around the organs for protection. It's a molecule that's really beneficial for long-term energy storage because it's very stable. It's very chemically stable. It doesn't lose energy in its storage process. Additionally, it doesn't have an impact on the osmolarity of the cell, and that's really how much water is coming in or out of the cell. Depending on the concentrations, water will sometimes diffuse in or out of the cell. The triglycerides, the fat, are not impacting this, and so it has no osmotic effect on the cell. It's also about 2 times more energy dense or efficient than carbohydrates in the process of respiration, so it provides a lot more energy than what a gram of carbohydrates would. It's also a port heat conductor, and so it's a good source of insulation, and we see this in marine organisms in cold environments, like a walrus, for example. It's got lots of blubber, helps it to stay warm. And then lastly, it's also, because it's a liquid at body temperature, our body temperature is much warmer than room temperature, it's a good shock absorber to protect the organs, kidneys, again, for example. The phospholipid molecule is also really beneficial and is crucial to living organisms because it's the molecule that helps to make up the cell membrane. And its 2 parts, as we've discussed. The phosphate head is hydrophilic, can be attracted to water, and the hydrophobic tails, the 2 fatty acid tails, are repelled from water. They don't interact. And so phospholipids are unique in that they have both portions. They have both a hydrophilic and hydrophobic, and this is called amphipathic, when a molecule has both hydrophobic and hydrophilic aspects to it, the phosphate head being hydrophilic and the fatty acid tails being hydrophobic. And so when these molecules mix with water, the phosphate heads are attracted to the water and the tails are repelled, and so they orientate themselves or self-arrange themselves into a bilayer where, as we can see in our picture, the phosphate heads are exposed on the outside to the liquid, and then the tails are protected, pointed to one another. And so this creates the phospholipid bilayer, and it's a really stable structure that helps to form the cell membrane that we'll look at in more detail in a coming unit. The last type of lipid molecule that we want to look at is called a steroid. And this is a group of lipid molecules that are similar to sterols, which are a type of unsaturated fat alcohol, and it's usually a waxy solid. And so steroids have some unique characteristics. They're 4 fused rings of carbon atoms, as we can see in the images here. They have 3 cyclohexane rings and 1 cyclopentanane ring. 17 carbon atoms in total in the rings. And so if you were given an image of this molecule, you would need to be able to recognize it. And the key there is that it's got the 4 fused rings of carbon atoms. Steroids are generally hydrophobic. This allows the passage through the phospholipid bilayer. They're usually hydrophobic, and so they can move across the phospholipid bilayer. There's 2 main types of steroids that we want to look at. Estradiol would be the first, and this is a hormone that's made naturally in the human body by the ovaries. It's crucial in regulating the menstrual cycle, cardiovascular system, neurologic system, skeletal system, vascular system. It has some big impacts on helping to regulate these different systems. It can easily pass through the membrane. It's a type of steroid that can pass through the membrane. It diffuses across the target cell plasma membrane and binds to the receptor, and then this receptor enters the nucleus, where it binds to the DNA and can help to actually regulate the transcription process of the genes. The second example of a steroid that we want to discuss is testosterone, which is a major sex hormone in males. It's essential to the development of male growth and masculine characteristics. It's produced in the testes, travels through the blood and carrier proteins, and it passively diffuses through cell membranes of target cells, so it's also able to pass through the cell membrane as well. And so these different molecules make up lipids and have these unique characteristics, as we've discussed. Next, we'll turn our attention to a second macromolecule, the protein.